Welcome to our final lecture video for the semester. So in this part, we will be kind of building on the stuff that we learned in the previous video about regulation of gene expression and apply it to two specific situations, development and cancer. So we'll start with development. So when we talked about things like albumin, which is expressed in liver cells, and crystalline, which is going to be expressed in the lens cell of the eye, we talked about how those situations can come about because there are different pools of activators circling around in the nucleus of those different cells. So again, if you wondered how different cells start out and end up with different sets of activators and other molecules, we can solve some of this by looking a little bit more deeply into development. So here you can see beautiful union of sperm and egg. Okay, and so what we're going to be focusing on here is really what's going on in our egg. So you can see here that there's some uneven distribution of molecules in the cytoplasm of this unfertilized egg. So we can see some orange circles, we've got some green triangles. These can be proteins, or they can actually be different types of RNA. Either way, they can get segregated to two different sides of the cell. So with this segregation of these molecules, these cells are beginning to gain momentum towards a specific fate as soon as fertilization occurs and the zygote divides for the first time. So check out this guy at the bottom. You can see that the distribution of our orange spheres is going to be primarily in this left-hand cell, that little triangle hanging out here. And then we've got our second cell on the right that's going to have these green triangles with just a couple of spheres. So just imagine that these are different pools of activators, and you can see how we're setting this up for a path to become different cells with different things going on, different cell types. Okay, so again, these can be proteins or RNA molecules. Whatever they are, they're unevenly distributed. Okay, so let's carry this a little further. And here you can see we have an early embryo, which is going to be composed of 32 cells at this point. So what we're going to be focusing on again is thinking about the sort of uneven distribution of molecules that we set up in the previous slide and what that's going to mean for this early embryo. So these guys at the bottom are going to have different molecules in their cytoplasm than the ones that are closer to the top. Okay, and as we get a larger embryo, of course, these differences will get more striking and we'll be able to start to develop different tissues and cell types. So as they continue down whatever path they're going to take, are they going to become an arm? Are they going to become a liver? Are they going to become a lung cell? They start to communicate with each other and these cells start to send and receive signals. So you can see signaling molecules coming from the sending cell down here to the receiving cell. And this starts to kind of reprogram neighboring cells so that they know to either, come on, we need more of this cell type, or alternatively, hey, I'm already becoming a liver cell, an eye cell. You need to become something else. This will also set us up for getting symmetry, like how we have an arm on the left, arm on the right, that type of thing. So we'll see some examples of that in Drosophila in just a little bit. So these signals will allow us to develop eventually definitive structures that are going to form in the organism and allow us to develop symmetry. So again, it can all start kind of snowballing from the beginning when you have that initial asymmetric distribution of molecules. Okay, so let's look at a molecular example first, and then we'll kind of zoom out and look at an example in Drosophila. Okay, so here we're looking at the path that a cell would take. So this is an embryonic precursor cell. The cell at this point has endless potential. It can become anything, but we're going to make it become a muscle cell. Okay, so there are a few things going on here. We have to think about what does it mean to be a muscle cell. So let's actually start over here on the right, where we've got this sort of collection of so-called other muscle-specific genes. So I'm sure you could imagine the types of things that need to be expressed in muscle cells. So maybe you have things that are going to contribute to the formation of the sarcomere. Okay, so maybe you're going to, have structural proteins that are going to hold those muscle cells together. These are going to be specific genes that a muscle cell needs to become a muscle cell. On top of that, we're going to have regulatory genes that help us turn on or off these turn on or muscle-specific genes or off other types of genes in the cell. So this is a master regulatory gene. This particular gene is called MyoD. Okay, so note in our embryonic precursor cell, MyoD is not expressed. The specific muscle genes are not expressed. The cell is not yet a muscle cell. It doesn't need these muscle-specific genes at present. But watch as we gain momentum toward becoming a muscle cell. So here's a myoblast. So a myoblast, this is kind of, in some ways, like a precursor to a full muscle cell. So what this means is basically this myoblast is determined, okay? Its fate has been determined. This is like when you guys say, okay, I'm going to go to medical school, but I'm not a doctor yet. Okay, so that's what's happening, okay? So molecularly, we can see what's happened to transition this is we've started to increase expression of MyoD. That was this master regulator. So this is a transcription factor. So this master regulator, hopefully you're imagining that we're about to have some effect on these other muscle-specific genes. Whoa, okay. So check it out. You don't have to be too worried about this. Just look, this is the pink gene from above, this is the orange and the yellow ones. MyoD can do a lot of different things, including sort of this positive feedback loop, making more of itself and increasing its own transcription. But more importantly for now, let's look at what goes on over here on the right. So our MyoD can start to increase transcription of this pink gene, which is yet another transcription factor, which can domino down to increase expression of these orange and yellow ones. So look, this can be things again like myosin, which we know is very heavily part of a muscle cell, part of that sarcomere. Okay, we can have things like cell cycle blocking proteins. Muscle cells are not big on dividing, right, because they've got all of that structural material that they can't really easily take apart and put back together. So we've got a lot of things that are going to be kind of the essence of a muscle cell. And then upstream of that, we have switches to turn these structural things on and off. So you can see, again, how this snowballs. So if you imagine back to those embryos that we looked at on the previous slides, you can see that having something that's going to trip this expression of MyoD towards being on, then you start this whole cycle, hey, I'll make even more MyoD, which will then help me turn on this pink transcription factor, which will then turn on these structural things. So the decision really gets made further upstream to be a muscle cell, and then we just start actually doing it down here once we get the pieces. So molecularly, that's a really kind of interesting example of how we can determine the fate of these embryonic precursor cells. We can also, again, think about how regulation of gene expression contributes to symmetry. So here, of course, is our Drosophila, because where would we be if we didn't do something to Drosophila? Okay, and of course they have symmetry. They have this dorsal ventral sort of thing going on. They've got our left and right. So of course we've got wings on both sides, 3 legs on both sides, 2 eyes, 2 antennae. So we've also got this anterior. One end has to know that it's going to be a head. The other end needs to know that it's going to be thorax, abdomen, etc. Posterior. So we have to be able to situate that. Okay. So we can see kind of how that starts to occur. And this is just a generalized view through development. And you can think about these different colors having different types of gene expression going on. So here we have our egg, okay? We're going to have the nucleus. This is within the ovarian follicle, so we're not even fertilized yet. Got nurse cells there to support it. Here we can see this is the unfertilized egg, more mature. We've depleted those nurse cells. Okay, the nurse cells are important for its survival, but not important to this concept. Okay, so then we have fertilization, beautiful union of sperm and egg. Okay, and the egg is laid. From here, it will start to become a larva. Okay, and you can see how having sort of asymmetric distribution of molecules in this sort of upper part could lead to this sort of symmetry and this anterior posterior situation happening in our larva and our adult Drosophila. Okay, so let's look in more depth at one of those molecules that can be asymmetric in our Drosophila. So here we're looking at the mature unfertilized egg. So this was that second stage we saw in the previous slide where we've depleted those nurse cells, but we're not fertilized yet. That's fine. Okay, you can see this is an actual image of the egg. This is, again, that illustration. This is going to be staining for a specific messenger RNA called bicoid. Okay, and again, pictured down here, but this is pretty obvious, even just from looking at this microscopy. Okay, notice after fertilization, and you got to really think about the scale here because this is an early embryo and a lot of cell division's happening. You can see individual cells now. We've transitioned from this one cell to many cells. That bicoid RNA was sequestered on the left-hand side. Okay, and so you can see now that on the anterior end, we still have a higher concentration in all of these cells that are on the left relative to the right. So things that are on the left end are getting the signal, hey man, I'm ahead. Okay, so this is where our anterior end is going to develop. All right, and you can see this is a real life gradient. Here we're looking at a single cell. Here we're looking at, you can see multiple cells. This is after some cell division, but that gradient still is retained. So what happens if we mess around with this? Well, scientists have, okay, and here's a regular fly, regular Drosophila. Note its eyes, it's got antenna, okay, its little mouth parts. And if you mess around with some of this gene expression, notice this. This is a leg, and here's another leg that are coming out from where the antenna should be. Okay, so for this guy, it's leg day every day. All right, he's got legs coming out of where his antenna should be. So you can really see how dramatic some of these systems are for positioning in our fruit flies and for other organisms. Okay, so this is a little bit of looking into how development works. If you're more interested in this, you should definitely consider taking our development course that Dr. Richardson teaches. But for now, we're just using this to illustrate why regulation of gene expression is going to be so important. Okay, so this is one example. All right, so our second example for this video is cancer. So obviously something that we're going to try and avoid for ourselves. Okay, so we're going to talk about two different types of genes. The first one on this slide is going to be proto-oncogenes. Okay, so proto-oncogene, we know oncogene, that's going to be related to cancer, but it's important to understand that a proto-oncogene is a normal gene. You should have these functioning in your body. Largely, these proto-oncogenes are genes that are related somehow to cell division and growth. So this would be like those receptor tyrosine kinases that we talked about that recognize growth factors that are going to be related to cell division. Okay, so proto-oncogenes are things that, again, are largely related to cell division, and you want that to be tightly controlled because cancer, really at its most basic level, is uncontrolled, inappropriate cell division. So you want to divide just enough and not become obnoxious about it. Okay, so there are multiple ways that a proto-oncogene can transition into a cancer-causing gene. So we can, anything that's going to give us more of the protein or overactivate the protein that we have can contribute to this. So what we're showing down here are a few examples. There are 4 examples, but really, again, anything that contributes to the increased production of this protein or the increased activity of this protein could contribute. So here, what we're going to have is actually what's called translocation transposition. Okay, what we really need to know about this is that this gene has been picked up from its regular habitat. Notice there's no real mention of a promoter here. It's regular habitat and dropped in a region under the control of a new promoter. And what we're talking about here is a stronger promoter, one that is more likely to allow transcription, giving us more than the usual product. Okay. We can also have a situation where the gene is copied. In this case, we've got now 3 copies instead of 1. Maybe these are going to be each, by themselves is expressed normally, but now you have 3 of them doing it all at once. So we have 3 times the growth stimulating protein that we had before. Okay, and then finally over here on the right, we've got the point mutations, one within a control element. This looks like a distal control element, okay, that allows for enhanced, increased expression, increased transcription. So this gives us more of the product. And then over here, a small mutation in the gene, the coding region itself, that causes this protein to be hyperactive. So this could be something like, again, if we're going to think about those receptor tyrosine kinases, this can be something, maybe it can mimic phosphorylation. So it seems like it's phosphorylated all the time, even though it actually hasn't been stimulated by a growth factor. So there are a lot of ways that you can arrive here, but these are some good solid starting points. Okay, so proto-oncogenes are the first category of genes that are important in cancer. Okay, and let's see one of these kind of in action. All right, man, so this is receptor tyrosine kinases. So you knew these would be important. So here we're going to have growth factor, okay, binding these receptor tyrosine kinases. Of course, they form the homodimers, they autophosphorylate, and then we've got activation of several things probably, but one of which is going to be a specific G protein. This one's called RAS, okay? Don't worry too much about that, but worry about kind of the downstream effects. It's activated, and we have, normally speaking, kinase cascade, phosphorylation cascade, that's going to activate transcription of whatever gene this is that will allow us to enter into the cell cycle. So all of this is normal. This is a normal thing that should happen when we need new cells. All right, the mutation that we're using as an example up here is in this box. So for this mutation, this is hyperactive RAS, okay? So basically, it's ready to signal without getting input from these receptors. So it's not the problem with the receptor. They're functioning normally. It's that RAS doesn't care what the receptor is doing. It's like, nope, just go ahead. I don't care if you didn't have a growth factor. Just keep dividing. Okay, so this can be one of the things that can contribute to cancer, okay, that's put into context. All right. So the second type of gene that we need to talk about is going to be a tumor suppressor gene. Okay, and so these are kind of biochemically speaking the opposite of proto-oncogenes. Where proto-oncogenes are going to be heavily involved with encouraging cell division, tumor suppressor genes are there to balance that out and inhibit the cell cycle. So again, what's happening in this main box is a normal pathway. So let's go ahead and introduce this protein. This is called P53. It's a transcription factor. This is an important guy, so probably just as well that you get used to him now, because you're going to see this a lot anytime that you talk about cancer or anything related to cell division. You're going to hear about P53. Okay, it's a very strong tumor suppressor. It's one of the main ones. Okay, so P53's job is really to kind of prevent inappropriate cell division. So there can be a lot of reasons that cells shouldn't divide. Sometimes it's because there are too many cells neighboring the other cell already. In this case, what we're looking at is kind of excessive DNA damage, so damage that's gone unrepaired. So you guys have talked about the cell cycle before. Before we ever even really get started into division, the cell does some checks. And one of the checks is for the kind of the state of its DNA. If the DNA is excessively damaged, it's not going to enter the cell cycle because it doesn't want to pass that DNA on. It doesn't want to try and replicate this if it's damaged, okay? So if it's fixable, it will go ahead and fix it and then reconsider entering the cell cycle. If there's just too much, this is where hopefully we're going to undergo apoptosis. Okay, so in this case, there's DNA damage. We're going to have some cell signaling going on here. This is just some kinases again. We sense DNA damage. We activate indirectly P53. It's going to act, again, this is a transcription factor here, and it's going to then say, hey, hang on, stop. Okay, so P53 is responsible for basically putting the brakes on. Wait a second, we've got a problem. The cell can then go back, figure out if it's going to fix this or what. In the context of cancer, if we're going to look, our mutation is going to be up here again in this gray box. All right, so if we have a defective copy of P53, if we have a defective kind of place where it can't bind to the DNA or it can't perform its function, or if it's just not produced, then we are going to have a problem, okay? So what would happen then? Of course, if we've got DNA damage, we're not going to have anything to put the brakes on. Yeah, okay, we may sense it, but we're just going to proceed through our cell cycle. Okay, so that can be a problem, all right, because this DNA damage can lead to further mutations downstream, which can again kind of snowball into larger effects. Okay, so the good news is that cancer is not caused from just one mutation. If it was, none of us would be here very long. Okay, but it's kind of the cumulative outcome of many problems. So what's shown on this slide is really just an example of the progression of one malignant growth. Okay, so it shows kind of the example. All right, so in this case, we're looking at colon cancer. So here's a normal colon epithelial layer here. The first mutation we might get is loss of a tumor suppressor. Okay, so this is another one called APC. There are other types of tumor suppressors besides P53. They can also do the job, but in this case, we've lost one called APC. This can result in maybe a little bit of cell division here that we didn't need, something, a benign growth, a tumor that starts to grow, maybe a polyp. Okay, so benign, it's not really a problem yet, but it's started down a path that's not looking good. Okay, so from here, if we let this go, okay, there's the possibility that it could accumulate further mutations, especially because we've removed one of the brakes on cell division. So if another mutation occurs, it's somewhat less likely that we're going to be able to stop cell division to deal with that damage. Okay, in this case, what's happened is we've gotten the overactivation of RAS, which was part of that oncogene, the proto-oncogene pathway we discussed a couple of slides ago. So this is going to push us, tip us in the direction of more cell division. Okay, step 3, we've lost yet another tumor suppressor. So we've kind of like added one to the gas, pushed on the gas a little harder. We've lost 2 of our braking systems here. So that's again putting us on a bad path. The more of these mutations you accumulate, it makes it actually even easier to end up with more, because again, those checks are not in place. So if we accumulate a mutation, it's more difficult for us to stop and fix it at this point. Okay, so next we can get here we've lost step 4. There goes P53, which hopefully now you've seen the significance of. We can get additional mutations. These other mutations can be even not shown on this slide, but you can relate this back to chapter 16, DNA repair mechanisms. If you get mutations in DNA repair, this can make it, send you down that path towards cancer. People with nucleotide excision repair mutations can end up with excessive amounts of skin cancer from normal sun exposure. So DNA repair, while not shown on the slide, definitively is part of this picture. Okay, and we also talked about the role that telomerase can play in sort of immortalizing those cells. Okay, so now we've gotten lots of mutations, kind of snowballed. Now we have a tumor. This small benign growth was somewhat playing by the rules. This was be like, you wanted to compare it to maybe going like 5 over the speed limit. Like, okay, it's doing something it shouldn't, but it's quasi playing by the rules. The more mutations this accumulates, the less it cares about the rules. One of the rules is, of course, kind of stay in your own territory for cells. And at this point, this malignant tumor has started breaking through its own territory and has become kind of part of this outer wall. And at this point, some of these cells can break off. And when they break off and move to another site, that's when you have metastasis. Okay, so again, this does not even really scratch the surface of cancer, but I think that this is a good example to relate this to, again, the importance of the regulation of gene expression for, our bodies and our cells in general.